healthy human body is an exercise in strict mechanical precision. Our survival relies on billions of microscopic engines operating continuously without a single misfire to keep our blood flowing and our organs oxygenated. Inside a single healthy cell, mitochondria act as perfectly contained power plants. At the very end of their energy production cycle, the electron transport chain, they cleanly reduce oxygen into harmless water. No leaks, no toxic byproducts. We can see this perfect machinery in action on this diagram. Looking at step 3, along the blood vessel wall, the endothelium, an enzyme called eNOS acts as a protective manufacturer. It binds L-arginine and oxygen to constantly produce a molecule called nitric oxide, or NO. Nitric oxide diffuses into vascular smooth muscle, keeping the vessels relaxed and fully dilated. It also coats the inner walls, preventing platelets from sticking together and ensuring smooth, unobstructed blood flow. Human life relies entirely on this chemical tightrope. Every engine, pump, and enzyme must remain perfectly tuned to sustain the body rather than destroy it. But severe physical trauma shatters that balance. When a patient suffers acute trauma, like septic shock or severe ischemia or massive crush injuries, the system drops into crisis. The immediate microscopic consequence is mechanical failure. The damaged mitochondria stumble under the stress, and highly charged electrons snap loose, leaking directly out of complex 1 and 3 of the transport chain. Referencing step 1 of our diagram, we see the disastrous chemical reaction that follows. An available oxygen molecule grabs this rogue electron, mutating instantly into superoxide, a highly unstable and dangerous reactive oxygen species, or ROS. A single misplaced electron has broken the machine. It lights a microscopic fire that turns the very oxygen we breathe into a cellular weapon. Once created, superoxide acts as an indiscriminate wrecking ball, launching a rapid three-front war inside the injured cell. First, it attacks the cell membrane. It strips electrons from the protective phospholipids, a process called lipid peroxidation. The compromised membrane begins to leak, sodium rushes in, and water immediately follows, causing the cell to violently swell and rupture. Second, it targets the DNA. Superoxide oxidizes base pairs and systematically breaks DNA strands, forcing the cell to trigger p53 activation and initiate apoptosis, programmed suicide. Third, it disables the cell's physical hardware. Vital structures like sodium, potassium, and Serca pumps fail entirely, flooding the cellular interior with lethal amounts of calcium. This chart outlines the ROS death loop. The initial burst of superoxide strikes its own mitochondria, causing deeper structural damage. The broken mitochondria leak even more electrons, generating a massive, self-amplifying wave of reactive oxygen species. A cell destroying itself from the inside is catastrophic, but the true systemic danger begins when this internal chaos breaches the cell wall and spills directly into the bloodstream. In step 4 of the diagram, we see the clash outside the cell. Escaping superoxide violently collides with protective nitric oxide. The collision instantly fuses them into peroxynitrite, one of the most toxic oxidants in human biology. Peroxynitrite has a specific, highly destructive target. Looking at step 5, it actively hunts down and oxidizes a chemical cofactor called BH4. This molecule is the primary stabilizer that allows the eNOS enzyme to remain in its functional state. Deprived of this cofactor, the eNOS engine is broken, stalling nitric oxide production. This triggers the flip. The crippled eNOS enzyme now dumps electrons directly onto oxygen. Instead of nitric oxide, it manufactures massive amounts of toxic superoxide. The exact enzyme designed to protect your blood vessels has been hijacked. It has been reprogrammed into a relentless poison generator. This is eNOS uncoupling. When we zoom out from the microscopic biochemistry to the macro level of the human body, the consequences of this flipped engine are devastating. With eNOS actively pumping out superoxide, the endothelial membranes lining the blood vessels are ripped apart. The vessels lose their integrity, leading to massive fluid leaks and catastrophic tissue edema. Deprived of nitric oxide, the vessels completely lose their ability to dilate. Platelets rapidly adhere to the damaged walls, triggering widespread, unchecked microvascular clotting that chokes off blood flow. This specific microscopic breakdown is the mechanical driver behind some of our most lethal medical emergencies. It is the engine, driving septic shock, acute respiratory distress syndrome, and traumatic brain injury. What doctors diagnose on the outside as rapid, multi-organ failure is the physical result of billions of betrayed enos engines uncoupling on the inside. This cellular mechanism also explains a terrifying clinical paradox. When a critically ill patient is suffocating, the most instinctual medical response is to give them oxygen, but doing so can be fatal. Administering high doses of pure oxygen to these patients floods the already damaged mitochondria with a massive excess of fuel. Because we enos enzymes are uncoupled, they cannot process this fuel cleanly. The excess oxygen simply provides more raw material for the broken enzymes to convert into superoxide, drastically accelerating the death loop. Life and death in critical care balance on a single misplaced electron. True survival requires intervening at the cellular level, stopping the biochemical betrayal before the engine flips.